Good morning, everyone. I welcome you all again for this new episode from TransConnect. This is February 2023. I am Dr. Mohit Chaudhary, and I have been accompanied with my colleague Dr. K. Jaya Gauri, who is going to talk about the second part of the basic fundamentals of immunology. The first part we had covered around a fortnight back, where we had discussed about the innate and the acquired immunity and the players involved in the same. Today, in fact, innate immunity was covered in details, and today we'll be talking about the acquired immunity in details. And then we also have a third part where we'll be covering the rest of the part of the immunology. So over to Dr. K. Jayagori for talking about the acquired immunity.
Hello everyone. So we'll be seeing about the basic fundamentals of immunology part 2. So before going into the video, we'll start with immune responses. So when an antigen is exposed to an organism, it develops an immune response to fight against the pathogen that has entered inside. So this immune response can be of two types. One is primary and the second is secondary. So when does primary immune response occur? When the antigen is exposed to the host for the first time. That is, when an antigen initially enters the body, what we encounter is the primary immune response. It is a function of the innate immunity. We saw in detail about innate immunity in the previous video. Now, usually what happens in primary immune response is that there is less number of antibody production and the rate of production is also low. The antibody usually produced in primary immune response is of IgM type. We'll see about antibodies in the further videos. The secondary immune response occurs when the same antigen is exposed again. That is when there is a subsequent exposure of the same sensitized antigen. This secondary immune response is mainly the function of the acquired immunity. In case of secondary immune response, there is large amount of antibodies produced. Also, there is faster production. Not only the quantity is increased, but also the speed of production is also increased. The antibody produced in secondary immune response is usually IgG. As we can see here, the y-axis indicates the blood antibody levels and the x-axis indicates the number of days. So when there is first exposure of an antigen, that is day 0, as we can see there is production of IgM antibodies, and then when there is second exposure, there is larger number of antibodies produced of IgG type. The secondary response is faster, quicker, and more amount of antibodies produced. Therefore, the antigen is chased out of the body faster than the primary response. We'll go into detail about the players of acquired immunity. The players of acquired immunity are mainly lymphocytes. They are either T lymphocytes or B lymphocytes. T lymphocytes are those cells that are produced in the bone marrow but are matured in the thymus. They acquire the knowledge of immunity at the thymus. That's why they are T lymphocytes. They receive inputs from various antigen presenting cells such as macrophages, dendritic cells, B cells, etc. After receiving inputs from the antigen presenting cell, they produce cytokines and thereby cause cellular destruction. B lymphocytes are those lymphocytes that are produced as well as matured in the bone marrow itself. They produce clones called plasma cells. That is, when an antigen is exposed to a B lymphocyte, it produces antibody against the specific antigen. The antigen producing B cell is cloned to form a plasma cell. Now this plasma cell will produce antibody only against that specific antigen. So lymphocytes are capable of recognizing specific antigens by means of genetic programming. That is, the B cells will undergo genetic rearrangement to produce a specific antibody. So when different cells identify different epitopes of an antigen, more than one antibody is produced for an antigen. That is, there is a B cell number A. It identifies an antigen at a particular site. It produces antibody against the particular antigen. Now, a single antigen can have many binding sites. For a different site, another B cell will bind, and the B cell from this, the antibody from the second B cell will attach to a different epitope. So this is where clonal selection takes place. For reagent purposes in blood banking, the best antibody has to be selected as all epitopes will not be equally reactive. Now this is the image that indicates genetic rearrangement. As we can see, there is 4 different domains. VH, DH, JH and CH. Each of these domains have different introns and exons. As we can see here, 1, 2, 3, 4, 5, and then mu, delta, gamma, etc. Now just imagine if the number 2 segment of VH, number 3 segment of DH, number 2 segment of JH and mu segment of CH binds, we get a rearranged gene. Now this gene will convert into mRNA and thereby convert into a protein, which is an IgM heavy chain. Now imagine the different permutation combination that can be done, producing different types of IgM. That is how individual IgMs are produced for the specific antigen. Lymphocytes, the lymphocytes that are mature and activated are called effector cells. These effector cells are the functional units of an immune system. So when does immune memory develop is that when these effector cells mature to form memory cells. These lymphocytes that become memory cells are permanently set to respond to the same antigen. These memory cells, they continuously circulate in our body even after the antigen has been chased out. The memory cells remain for a lifetime in the host. Now this memory cell is responsible for the faster, quicker and speed response of the secondary type that is encountered when the same antigen is met. Now T helper cell. T helper cell is a very important cell that forms the basis of the adaptive immunity. Now when an antigen presenting cell, be it a macrophage or a B cell or dendritic cell, when they present the antigen to the T helper cell by means of major histocompatibility complex. That is, for macrophage it is MHC1, for B cell it is MHC2, dendritic cell has both MHC1 and 2. So what the antigen presenting cell does is that it engulfs the antigen by phagocytosis, breaks the antigen into pieces and presents the part of the antigen to a T helper cell, telling that so and so has come inside to invade us, like that. Okay, so the T helper cell 1 activates macrophages to perform phagocytosis and kill the cell. It activates cytotoxic T cells that are destructive T cells, encouraging them to destroy the antigen and the cell containing the antigen. So T helper cell 2 activates the B cell and stimulates proliferation and differentiation of the B cell into plasma cell. That is a cloned B cell that produces one type of antibody. As we can see here, the antigen presenting cell engulfs the antigen, breaks the antigen into pieces. By means of MHC, presents the antigen to the T helper cell. The T helper cell, which gets activated, stimulates the B cell to get itself cloned into plasma cell that produces antibody against the antigen that was presented initially. The T helper cell also stimulates the cytotoxic T lymphocyte to kill the cell. So coming to antigens. What are antigens? Antigens are nothing but proteins that are part of or whole of a foreign substance or a pathogen or even abnormal self element that induces immunity or production of antibodies that bind to them. There is another entity called immunogens. Immunogens are nothing but any substance that elicit an immune response. Not necessarily it has to bind to an antibody. So there are another entity called haptens, that is low molecular weight antigens. The antigens that are usually less than 40,000 mW are called haptens. They become immunogenic when they are attached to a carrier protein. That is, when they bind to the carrier protein, the molecular weight of the hapten is increased. Thereby, they are capable enough to perform as an antigen as a whole. So the term epitope indicates that the site of the antigen where the antibody binds. These are a few terms that we are supposed to know regarding antigens. So most of the antigens are usually found on the surface of the foreign cells or damaged self cells. So what are the factors that influence an immune response pertaining to an antigen? Number one is the size. The larger the size, the better the immune response. Number two is the complexity. That is, a more complex antigen will produce multiple different antibodies for the same antigen. Next thing is the conformation. D isomers of antigen have better conformational, are better in conformation and better in producing immune response. Charge. The more negative charged antigens are found to be more immunogenic. Accessibility. If the antigen is not exposed to the MHC containing antigen presenting cells, then the antigen will not be presented to the T helper cell and thereby immune response will not be there. Next thing is solubility. If the antigen is more soluble, they produce better immune response. Next is digestibility. An antigen presenting cell has to engulf the antigen, break down and then present it. So if the digestibility is easier, then there will be better immune response. Next thing is the chemical composition. Like proteins are better antigens than lipids, which are better than carbohydrates. Next thing is the complement system. Complement system are nothing but proteins. They are present in the precursor form throughout the circulation. They get activated at sites where inflammation occurs. Complement system is known to be the bridge between innate and acquired immunity. The complement system has mainly three functions. One is the final lysis of pathogen. Number two is opsonization, that is coating of the antigen, and phagocytosis, that is engulfing of the antigen. The next function is inflammation. So the complement system has three different pathways. One is the classical pathway, two alternate pathway, and three is the lectin pathway. The classical pathway is activated by antigen antibody binding. The alternate pathway is activated by the polysaccharides of a pathogen. The lectin pathway is activated by mannose binding lectin. So this is a pictorial representation of the three different pathways of complement system. As we saw, the first pathway is the classical pathway, which will get activated by antigen antibody binding, by amyloid substances, by apoptotic cells that have been destroyed, and by some neuronal blebs also. So the classical pathway begins with complement C1. The C1 the C1 is converted to C3 by the help of complement 2 and complement 4. C3 is an important complement that further gets broken down to C3A and C3B with the help of the enzyme C3 convertase. C3 convertase enzyme is nothing but the combination of C4B and 2B. Now this C3A plays an important role in chemotaxis and activation of WBCs. The C3B plays an important role in opsonization, that is coating of the antigen. The C3B combines with the C4B2B. Now C4B2B and 3B together is the C5 convertase. C5 convertase enzyme breaks down C5 into C5A and C5B. C5A is also an important complement that performs chemotaxis and activation of WBCs. The C5B binds with the complement C6, C7, C8 and C9 to form the MAC. We will come to MAC later. Now the lectin pathway. Lectin pathway is activated by the mannose binding lectin. When the mannose binding lectin activates a serine protease, that is mannose binding lectin associated serine protease, it also converts C1 to C3, thereby producing C3. From C3, the same pathway continues, that is C3A and B is broken down. C3B combines with C4 and 2B to form C5 convertase, producing C5B and thereby producing MAC. The next pathway is alternate pathway. In alternate pathway, what happens is when the pathogen's surface is exposed, there happens a hydrolysis, that is water mediated breakdown of C3. Now this water mediated breakdown of C3 forms a hydrolyzed product of C3. This hydrolyzed C3 combines with factor B and factor D, producing a C3 convertase. This C3 convertase breaks down a whole C3 into C3A and B. C3B proceeds for opsonization and forming another C5 convertase made up of C3B, factor B and 3B. So two compounds of C3B and factor B combines to form another C5 convertase. This C5 convertase also breaks C5 into C5A and C5B. C5A proceeds for chemotaxis and WBC activation, and C5B combines with C6, C7, C8 and C9 to form the MAC. The MAC is nothing but the membrane attack complex. This is nothing but what happens is when a cell is infected by an antigen, or if the infective cell is present, or any cell for that matter is pathogenic, then this membrane attack complex penetrates the cell surface, forms a hole in it, causing influx of all water, other proteins, etc., and thereby causing cell lysis. So ultimately, the complement pathway ends in the formation of membrane attack complex producing cell lysis. Apart from that, in the pathway there are various complements such as C3A and C5A that is responsible for chemotaxis, that is attracting various cells, and leukocyte activation, that is activating the WBCs, and opsonization, that is coating of a pathogen. So in the next video, we'll be seeing all about antibodies, the types of antibodies, antigen-antibody interaction, and there will be a subsequent video for monoclonal antibodies and hybridoma technology. Thank you so much.